students, our topic would be Pentose Phosphate Pathway. We have already done with the mechanism of respiration, both anaerobic and aerobic respiration. In aerobic respiration, we find that there is glycolysis, then the pyruvic acid produced during glycolysis enters the mitochondria where it gets converted into acetyl coenzyme A, and then it enters into the Krebs cycle. And during Krebs cycle, we find that there is formation of reduced coenzymes, and the reduced coenzymes then forward their electrons and protons to the oxygen, which is the terminal oxidant, during the oxidative phosphorylation. And during this process, ATP is formed. But there is an alternate pathway for this respiration. In many organisms, a pathway occurs in the cytoplasm, and in the cytoplasm it takes place in presence of oxygen. So during this phenomena there is direct oxidation of glucose, and this is called the direct oxidation pathway also. This pathway was indicated very earlier in 1930s by a Nobel Prize winner physiologist Farooq. Later on, a little was done by Dickens on this pathway, but it is full mechanism, reaction details and reaction mechanism was worked out by Horicon and Raker independently. Now strictly coming to this pentose phosphate pathway, because we are not having much time, this would be a short video. We will start how the first step of this pentose phosphate takes place. In the first step, the glucose present in the cytoplasm, it gets phosphorylated and becomes the glucose 6-phosphate. We here need 6 molecules of the glucose. They get converted into 6 molecules of glucose 6-phosphate in presence of the enzyme hexose kinase. So the first step of the pentose phosphate pathway is that there is the phosphorylation of glucose to produce glucose 6-phosphate. Now the 6 molecules of this glucose 6-phosphate get converted in presence of the enzyme. This is an actually oxidation process. Dehydrogenation takes place. The enzyme is dehydrogenase. This is hexose phosphate dehydrogenase. It converts glucose 6-phosphate into an intermediate. The intermediate is 6-phosphogluconate or glucono-gamma-lactone. This is the intermediate compound, usually formed for a brief time, and immediately it is converted by the enzyme lactonase. In presence of water, it gets converted into 6-phosphogluconic acid. Now during this step, that is dehydrogenation of this glucose 6-phosphate pathway with the help of this dehydrogenase enzyme, here 6 molecules of NADP, this is nicotinamide adenine dinucleotide phosphate. Remember here NADP is used. Instead, while we were discussing the Krebs cycle, we used NAD positive, that was is another coenzyme. Now this coenzyme A, this is the NADP positive, oxidized, it gets reduced to NADPH2. Simply we will call it NADPH2, but actually it is NADPH plus H positive, which we have earlier discussed why it is so. Now here, during this step, there is dehydrogenation. Now, in the next step, this 6-phosphogluconic acid is again oxidized. That there is once again dehydrogenation, but along with this, actually there is oxidative decarboxylation also. So here we can say the enzyme is 6-phosphogluconic dehydrogenase and decarboxylase, because this enzyme is both a dehydrogenase enzyme and a decarboxylase enzyme. During dehydrogenation, it gives these hydrogens to the NADP positive, which gets converted into NADPH2. Now we will call it NADPH2 instead of NADPH plus H positive for the simplicity. Here also 6 molecules would be used and 6 molecules of NADPH2 are formed. Now during this process, there is decarboxylation and 6 molecules of carbon dioxide are evolved. So the reaction results in the formation of 6 molecules of ribulose 5-phosphate. So here we can say initially we were having a 6 carbon compound. Now from each molecule of 6 carbon compound we have found here there is removal of 1 carbon in the form of carbon dioxide. So from 6 molecules of carbon dioxide are removed. So we will have now 6 molecules of a 5 carbon sugar, that is ribulose 5-phosphate. Now this ribulose 5-phosphate 6 molecules, they get converted into their isomers. Two types of isomers are here formed. One is ribose 5-phosphate. Remember, this is the very important significance of this pathway, that during this pathway these ribose sugars are formed, which are very important. They act as precursors of nucleic acid synthesis. And also one more isomer of this, that is xylulose 5-phosphate. So xylulose 5-phosphate and ribose 5-phosphate. You know we have 6 molecules of ribose 5-phosphate. Now 2 molecules of this ribose 5-phosphate are formed in presence of the enzyme isomerase, and 4 molecules of this xylulose 5-phosphate are formed in presence of the enzyme epimerase. So let us have a little recap of this.